So hello and welcome to this session. All of you can hear me properly? All of you can hear me properly?
Yes, we can hear you.
Great. So it's time, I guess, 7:30 p.m. So today's workshop is a 2-hour special workshop. We had some, difficulties at our end. That's why we had to shift yesterday's workshop to today instead of September 10th. Today is September 11th. And before we start, I guess all of you have covered your done your part at the educational platform. So those of you have uh, are yet to join, let us wait for a few moments. Maybe we have 12 participants here. So what we have, okay, Apar, thank you very much. Shall we wait a little bit? Let's wait for a little bit of time so that everyone can participate and join in. Yeah, so let's start, I guess, because let others join in. So those of you who have joined, the first thing I would like to tell you about is review what we have done so far. Can you guys hear me?
Yes, Pratim.
Okay, shall I start?
Sure, please start.
Okay, thank you very much. So review of whatever we have covered so far as a part of this data science for biologists program. We started in 20th of August and we covered topics like introduction to big data bioinformatics on August 22nd, where we learned about knowledge-driven versus data-driven discovery and showed you some examples about the applications of bioinformatics in agriculture, neuroscience, and cancer biology, immunology, and so on. And also we showed you some data repositories, specifically on cancer, metagenomics, and also transcriptomes and all this sort. Thereafter, we moved on into genomics, where we understood about the basics of molecular biology like cell nucleus and chromosomes, and thereafter DNA molecule and structures and genomic variations. And we also understood what actually we mean by targeted sequencing, whole exome sequencing, and whole genome sequencing. And we did some analysis using the tBioInfo server, uh, and we tried to understand what are variants, how a variant calling is being done, what do we understand by variant, differential variant calling, and so on and so forth. Thereafter, we moved on to transcriptomics, and we understood about why is it necessary to process raw NGS data, and how is it being quantified, what we actually understand by quantification, And we did some differential gene expression and factor regression analysis. And we also told you about the resources that you might use for interpretation using gene ontologies and gene set enrichment analysis, although we did not go in much detail about that, because I know these are things which require a little bit of expertise on your part, for which we always recommend you to go back to the educational portal, edu.t-bio.info, and do the necessary courses out there, necessary modules out there pertaining to genomics, pertaining to transcriptomics 1 and transcriptomics 2. Thereafter, we moved on to transcriptomics 2, where we understood about quantile normalization technique, how to remove noise, what is log transformation, why is it necessary for log transformation. We understood something about kurtosis, and thereafter we looked at the data and we told you bits and pieces about PCA, principal component analysis. How is it important for reducing the dimension of the data into some form where we can visualize it into two-dimensional, into two dimensions to be very specific. And we understood about outliers, filtering, and normalization at the same time. So today, the progress we have so far in the course, this is the progress till date. So I can see some amount of activity and some amount of activity in the educational profile, in the educational platform, and also in the server at the same time. And this is the organization page where you can find all the details about the programs, starting from which program is going to take place, which class is going to take place on which date, and also the resources that the recordings of the lectures. So some of you were asking in the WhatsApp group today, I remember, that where can you get all those recordings. We regularly update those recordings with a lag of 1 or 1.5 days to the forum itself, the forum tab in the organization page, where you can find the lectures, recorded YouTube videos of the previous day's lecture. And this organization page is very important, because during the entire length of the course, whatever is the exchange, whatever materials that you need to study or you need to kind of get informed with, will be there in the forum page. So home, forum and progress, these are the things that you should. And also home, forum, and curriculum are the three main tabs you would see at your end. The home will be telling you about the upcoming thing, what is there, what is coming in the next week, and the forum is going to tell you about the resources, the previous day's lecture, and some reading materials, which are additional reading materials which we share with you, and the curriculum will tell you the entire length of the syllabus, which we are going to cover in the span of 3 months. Okay, so going forward, so first of all, I would like to, this pipeline we ran the last time when we did genomics pipeline. This was a demo pipeline, but we visualized the thing. I saw many of the participants here did some visualization, did run some visualization using the genomics pipeline. Nidhi is one of them, probably Ruchika is one of them, Jyoti or one of them. So the first thing I would like to tell you is that in this pipeline, we use two algorithms, basically Bowtie2, and we use something called visualization. As a part of visualization, we have used, as a part of the platform, there is something called JBrowse. So the people who are right now in their system, I would ask you to log in to the server right now, and therefore we can move along from there. We will kind of do it together. So I will take a pause here and transfer the stage to all of you participants. So So there is something on the chat. Let us see what is there. Yes, apart. Thank you very much. Yes, yes. So these are the places which you should be looking for. And so all of you who are in front of your system, your desktop or your laptop, it might not be possible over mobile phone. So maybe some of you are traveling back home, and but don't worry, if you can get back home by 8 or maybe 8:30, you still have time to. Or if you are not able to, um, log on to the system, you can do it after the class is over. So those of you are in front of your system, laptop or desktop, please log on to the server, and why don't you show us what you are doing right now. So if I may request, uh, start with in alphabetical order. Maybe Abhishek, would you like to share your screen? So I will stop sharing the screen. Those of you who are in front of your system, please if you want, you can write in the chat that I want to share my screen. So Abhishek, do you have are you in front of your system? Okay, don't worry, Sunita. This section will be recorded, so you can go on and do it. Don't worry about that. So Abhishek, going in alphabetical order, Abhishek, I am not getting a reply from Abhishek. What about the rest? Arvind, are you in front of your system? I would like to see that how do you run the pipeline. So this is, as I said, that this is a workshop where we like to see the screen. So once you say yes, I will stop sharing my screen, and you can do it at your own end. I would show you. We can help you out to share your screen. Arvind, are you there? Or maybe Devanita, maybe Devalina, Diksha, Jyoti, Prabir, Redmi. I don't know. Maybe I don't know
I
anybody.
am here.
Hi.
Jyoti is here.
Jyoti.
Yeah.
So great, Jyoti. Thank you very much. So I will stop sharing my screen. If you are in front of your system, you can find a button like this. Something like this. When you open the screen, there is a share button on the bottom. Did you find it?
From left top one?
No.
From where I have to log in.
So once you have logged in, you will see a window where my screen is being projected right now. So on the left, there is this, on the below panel, there is something on the left you find, there's a microphone, there's a video. After that, there are several buttons, and there are something called share. Can you find it?
Okay. Yeah, I am trying for that.
Did you find the share button?
Yeah, share button. From last which number? 1, 2, 3.
Probably. Let me stop sharing and say you. So on the left, you can find something called mute. If you click it, it will be muted. And then there's a start video button, and then there's another one called invite, then participants, and then something called share, a green button called share.
Okay, share. Yeah.
Yeah. Can you share your screen right now?
Yeah, the screen is visible for me.
Yes. So it will be visible for us.
Yeah.
Please share your screen and go to the server. And if you have the presentation open with you, then you will be, yes, thank you, Jyoti. Nice. Yeah, we can see it. So can you go to your server?
Where is server?
The server address is server.t-bio.info. Can you open a new? Yes, I will.
New window. Okay.
Yeah, you have to open a new window. The address is https.
Yeah. Wait for some time.
Sure, absolutely. Yeah. Take your time. https dot. See, I am pasting the link in the chat if you can find it.
Okay.
Go to the chat. You go to the chat. Copy the link and paste it out there. Open the Zoom. Open the Zoom. No, open the Zoom button that I can see on the right. Beside your, beside your,
Pratim,
yes.
let me guide her. Let me guide her. Pratim.
Yeah, sure. Why not? Go on.
Jyoti, can you minimize this? Can you minimize this one?
I got the Zoom group chat. https edu.t.
No, you need to log on to the server, Jyoti.
This is the educational platform. So the link is there in your meeting button. You go to the meeting where we are having the meeting.
Yeah.
Go to the software below.
On the lower panel, besides, yes, not this one. No, not this one, Jyoti. I can see the Windows button where एकदम नीचे जो है ना, एकदम नीचे आपके आपके कंप्यूटर स्क्रीन पे एकदम नीचे Zoom वाला जो दिख रहा है नीचे, Zoom का जो आइकॉन दिख रहा है
नीचे एकदम जहाँ पे क्रोम का बटन है ना नीचे.
Okay, so can you type the server address if I dictate to you?
Yeah, I am trying to open new windows from where I can got it.
No. You just open a new window. Just listen to what I am saying. Open a new window. Open a new window.
Yeah.
Open a new tab.
Yeah. Done.
Have you opened it?
Yeah.
No, you haven't. I can see your screen. You are on your Gmail screen right now.
Yeah. I am on Gmail screen. From there, I can do it.
Yes, of course.
Yeah.
Can you see a plus sign on the top? Can you see a plus sign on the top?
Open a new tab. नया एक खोलो ना, नया खोल दो उसमें एक.
ये देखिए प्लस करके एक साइन है ना ऊपर.
अरे प्रॉब्लम ये है कि ये लास्ट में यहाँ पे आ गया है. अब हो जाएगा.
कोई बात नहीं. आप उसके ऊपर राइट क्लिक कीजिए.
चलो हो गया. न्यू विंडो.
इसमें लिखिए सर्वर ACRVER.
ओके.
सर्वर डॉट T for Telangana hyphen BIO .info. Correct. Can you press enter? Enter, yes. So do you have the server login with yourself?
No, I never did it.
Okay, can you go back to your mail and get your server login?
Yeah, I am on mail.
Yes. So
from
you have
today's link.
No, this is today's link. But what you need to do is probably we have sent an email to you with server credentials. Can you send her the login credentials in email once again right now?
You can tell me the date. I can find from that from my mail.
So there is a search mail. No, there is a search mail.
Yes.
Yes. Write Mohit, MOHIT Mohit.
Oh.
So the mail would be from Mohit Mazumdar.
Or maybe server credentials here. The credentials.
Okay. Go down.
Please tell me the date. From that I can.
We don't exact. Yes.
22. Yeah. Credential is here. Credential and.
Yes. Can you go down? Yes. Scroll down. You will find the. Yes. So this is your username and password. So copy the username. Yes. Copy the username. No, not like this. Not like this. You just select the whole thing.
Yeah.
No. Okay. You just copy the email address from here. Don't worry.
Yeah. Copy the email address.
Yes. And now go to the server, the link that you entered. Yes. So log in. You can see a login button on the top right. Yes. So paste the thing here. Exactly. So now go back to your email and copy the password.
Actually, I had tried for that, like, but maybe I am not able to success.
Let's see. We'll help you out. That's why we had this session today, so that we can help individual people to log on. Yes. Copy that and paste yes, paste it out here. Excellent. Now log in. Press login.
Remember me. Okay.
Yes, Mohit?
No, I thought I will ask her to remember. But yeah, login is successful. Very nice.
Fantastic. See, login is successfully logged in successfully. Fantastic. So can you maximize your screen?
Yeah, it is already.
Fantastic. Okay, so now the pipeline that I was showing you was in respect to, so you can see 4 panels out here, right? NGS data,
Yeah,
mass
and this
spectroscopy,
data, yeah,
structural biology, and data integration and modeling,
I
right?
can see that.
So you have to, whatever we are doing right now would be connected to NGS data. So scroll down. Yes, scroll down a little bit. You can find something called mutational variant, parallel analysis of mutation variant data. Yeah. Click on this arrow. You can see a demo, variant calling cell line.
Yeah.
Yes. So can you click on that demo? Yes. Click on that. Excellent. Now you see you have a pipeline in front of you. Don't click on anything. Just scroll down. Make the whole pipeline.
I can see the pipeline.
Excellent. So now we have to build the pipeline from the scratch. So, Jyoti, this is a AI guided pipeline, which means that the very next button that you have to click. See, there are so many things out here, right? You have data type, data preprocessing. There are blocks which says data preprocessing. There are blocks which says mapping. There are blocks which says variant calling, and there are blocks which says differential variant calling, annotation, post processing, and segmentation, and so on and so forth. But if you look at the entire set of boxes that are here, the button which is highlighted, which is the highlighted button out here? Start.
Yeah, highlighting is in yellow color.
Yes, the highlight is in
Symbol
yellow.
as like top.
So which one is the button which is highlighted in yellow? Start button.
Clean printer.
Yes, there's a start button on the right top left.
Yeah.
Yes, so click on that. Click on that.
Yeah, done.
Yes, so this is the educational pop-up. So see, if as a part of the educational pop-up, so it says, what is this pipeline going to do? See, we are trying to detect mutational variants, and you see you can scroll down on this window. Yes, exactly. See, this is the purpose of this pipeline. See, so we are going to analyze genomic data here, and it says what are the blocks for. There are several blocks starting from pre-processing to mapping to variant calling and differential variant calling, and then annotation and post-processing and so on and so forth. So scroll up once again. So now we are going to build the mutational variant pipeline. So what does this pipeline do is explained here in this educational pop-up. It is meant for biologists like you who does not really have that bioinformatics background but wants to learn bioinformatics, right? So that's the whole purpose of it. So now I'm trying to guide you. So did you understand what I said?
Yeah.
Perfect. So can you scroll up?
Done.
No, yeah. So if you scroll down, you can find so many things out here. So once the class is over, yes. Yes, so we covered, if you remember, we covered all of these in our genomics class. This variant calling, different variant calling, what are non-model genomes, what are model genomes, etc., etc. Remember?
Yeah.
Excellent. So can you scroll up on this window again? Perfect. So now the immediate button, oops, wait for an hour, wait for an hour, click the wait for an hour, yes. So can you go up? Yes. So there is this OK button, yellow colored OK button. Can you see it? Click on that. Great.
Nothing is happening.
Oh, wait, it will take some time. Don't worry. Something will happen. Don't worry. So if nothing is happening right now, which means the server has not received any response from you for a long time. So what you should do, probably, you have to refresh this page. How to refresh this page? You can see on the top, there is a back arrow button on the top where the server address is written, server.t-bio.info/pipeline mutational variant demo pipeline. आपको दिख रहा है ना ऊपर एक बार एकदम लेफ्ट में, लेफ्ट, go to the left.
In main page?
हाँ, इसी पेज में है, इसी पेज में. हाँ, correct. So start again.
Start. Where I have to click?
ये देखिए एक स्टार्ट वाला बटन है ना, येलो कलर्ड स्टार्ट बटन. ये देखिए आपके लेफ्ट में. नो नो नो, वहीं पे आपके विंडो में ही है. स्टार्ट म्यूटेशनल वेरिएंट पैरेलल एनालिसिस ऑफ म्यूटेशनल वेरिएंट के जो लिखा हुआ है उसके इमीडिएटली नीचे एक स्टार्ट. हाँ, यस, क्लिक ऑन दैट. Click on that. Yes. Now press OK again. Right now.
Again, nothing is happening.
Okay, it might be so that the server is not getting any response from you. So you have a button in your keyboard which says F5. Can you see it?
Yeah, F5.
Press that F5 button.
I am pressing, but again nothing is happening.
It seems that the server is not happy with you. Don't worry. So what you should do is you go to the place where you've written the server address. Can you see that ऊपर वाला जहाँ पे आपने server.t-bio.info लिखे थे?
Yeah.
हाँ, उसके थोड़ा लेफ्ट में जाइए, उसके थोड़ा लेफ्ट में जाइए. लेफ्ट देखिए एक सर्कुलर बटन है. सर्कुलर नहीं, वो नहीं. उसी विंडो में लेफ्ट में एक देखिए बुकमार्क्स करके लिखा हुआ है. लेफ्ट में, लेफ्ट में, लेफ्ट. उसके ऊपर बुकमार्क के थोड़ा ऊपर देखिए एक गोल सा एरो वाला है. एरो बटन है ऊपर.
Just wait. In that page?
इसी पेज में मेरे को दिख रहा है. देखिए मैं दिखाता हूँ. ये देखिए जहां पे आपने सर्वर एड्रेस लिखे थे ना, जहां पे आपने सर्वर एड्रेस लिखे थे. हाँ, नहीं, बुकमार्क नहीं. बुकमार्क के ऊपर देखिए एक गोल बटन है, गोल एरो वाला. यस, दिस बटन. प्रेस दिस वन. Yes, press this one. Correct. So this is the refresh button. So often what happens, refresh press किया आपने?
Yeah.
Right. So press start once again. Okay करिए. See, now go down a little bit. हाँ, देखिए अभी देखिए बहुत सारा येलो बटन हाईलाइटेड हुआ है.
Yeah.
देख रहे हैं?
With symbol top.
Sorry, Jyoti.
With symbol top, that is your official symbol, you can say.
हाँ, you can. अब देखिए PCR clean वाला जो बटन है वो हाईलाइटेड हुआ है. बोटाई 2 हाईलाइटेड हुआ है.
PCR clean, botai 2. Yeah,
right,
I can see that.
Right, exactly. So the pipeline that I'm going to show you, you are going to do right now is botai 2. Press the botai 2 button. प्रेस कीजिए, क्लिक कीजिए उसको. हाँ, देखिए नया एक विंडो खोला है. ओके करिए, ओके करिए. Right. See, there are now the next button which is highlighted is visualization. देखिए उसी के नीचे एकदम मैपिंग. Yes, click on that. Right, okay, correct. देखिए अभी एक ही बटन हाईलाइटेड है, एंड वाला बटन राइट में. ऑन द राइट, एकदम राइट में नहीं, ऊपर. go up. Yes, click on that. See, now your visualization pipeline is complete. आप थोड़ा नीचे जाइए. See, this is the pipeline that you run. अभी जो pipeline आपने run किया, this is the pipeline you ran. इसमें आपने क्या किया? इसमें कुछ भी नहीं. इसमें आपका जो होल जीनोम सीक्वेंस फाइल है, उसको लेके आप बोटाई2 यूजिंग द बोटाई2 एल्गोरिथम, हाँ बोटाई2 एल्गोरिथम को यूज करके आपने रेफरेंस जीनोम के ऊपर यू अलाइन ऑन टू द रेफरेंस जीनोम, ठीक है। एंड नाउ यू वांट टू सी, नाउ यू वांट टू विजुअलाइज, आप इसको क्या विजुअलाइज करना चाहते हैं? यू वांट टू विजुअलाइज, लेट्स से यू वांट टू विजुअलाइज द वेरिएंट्स, वेरिएंट्स मतलब म्यूटेशनल वेरिएंट देखना चाहते हैं, व्हाट आर द म्यूटेशंस दैट आर प्रेजेंट आउट हियर, राइट। सो अभी देखिए पाइपलाइन, एक जहां पे आप स्टार्ट बटन जहां पे है ऊपर, स्टार्ट बटन, उसके ऊपर एक ब्लू कलर का लिखा हुआ है विजुअलाइजेशन, ऊपर ऊपर, यस थोड़ा सा, यस क्लिक ऑन दैट, सो इट विल ओपन अ न्यू विंडो। न्यू विंडो में क्या खोल रहा है? न्यू विंडो में देयर इज एन एप्लीकेशन व्हिच इज ओपनिंग व्हिच इज कॉल्ड जे ब्राउज। नाउ जे ब्राउज इज एन एप्लीकेशन व्हिच इज कॉल्ड genomics viewer, ठीक है। So genomics viewer will tell you that what are the variants out there. So I don't know why it's not opening. ना थोड़ा सा टाइम लगेगा, correct, yes. So this is the thing where we wanted to go. Now this will take some time to load. Now what I would do, request you that you just take your time, stop sharing your screen so that other people can, I can also show the other people how to do it. Is it okay?
Okay.
So you can, I will ask you to share your, yes Jyoti. So stop sharing, so excellent. See, I think all of you have got some idea. Diksha, yes, Diksha, can you share your screen so that I can show you? Devalina, yes, excellent, Devalina. So Devalina is alphabetically the next person. So Devalina, can you share your screen so that we can see it, and obviously unmute yourself? Devalina. Devalina, are you there? Don't worry, Sunita, we will help you out. Devalina, are you there? Excellent.
Can you hear me?
Who is this, if I may know? Devalina. Hi, Devalina. So, yeah, can you share your screen, Devalina?
Yes, I'm trying. Actually, here it's showing a window and application you want to share.
देखो, नहीं. If you are using Zoom, if you are within Zoom,
yes, sir.
The place where you can see, where you saw the screen right now, there is a button called share, which is colored as green.
Yes, sir. I have to click it.
Yes, click on that.
And so then I get another screen which is telling me to select a window and application that you want to share.
So select the default one. Select the default one.
Sorry, sir. Sorry, hello?
Select the default one. So share button, you press the share button.
So there are many options here. Like
which
screen.
one do you want to share?
So the screen I'm clicking on share.
Yes, click on that. Don't worry. Yes, perfect. We have not been able to see it, but we'll see it shortly.
So is it visible?
I think it will take some time, but let me see. Yeah, we can see it. Devalina, we can see your screen. So you are on the server platform right now, correct?
Yes, sir.
Excellent. So today, what I was telling Jyoti, we would be like to show you the visualization. So I think you have already understood what we are trying to do here.
Yes, sir. I have to go to mutation variant.
Yes. So so in genomics, the thing that we studied here is basically mutational variant parallel analysis of mutational variant data, right? So we have a demo pipeline. But why this demo pipeline? Because you need to understand that how the analysis and what sort of pipelines are being run. So let us visualize the data that we have right now. So click on that arrow where you, yes, click on the demo pipeline. So I think you know, just go around. I will guide you. Yes.
I think the connection is a bit unstable.
Don't worry. Don't worry. It will take some time, but don't worry. Don't worry. it will work. It'll take some time, but it will work. Don't worry. That's why we have fixed 2 hours today, because yes, exactly. So see, we have always been saying that this is an AI guided platform, which
Yes.
means that it is intuitive. So intuitively, the button that is being highlighted right now is start button. So click on start. Excellent. Yes. What happened?
Sir,
Why did you come out?
sir, I don't know. Just the screen just went.
No, don't worry. Do it once again. Excellent. Yeah. See, okay, exactly. So this is where we should be going. We'll be clicking OK. So the immediate pipeline that is visual. So see, there are two pipelines which are highlighted, two algorithms which are highlighted. One is PCR clean and one is BOTI2. I think, do you want to run the visualization or do you want to run the PCR clean?
So visualization.
Excellent. Click on BOTI2. Right. So next time when you do it, read the, whatever is being there in the educational pop-up. That will help you to make your life easier, and it would help you to do the project when you do it at the end of this course. Okay.
Yes, sir.
Okay. Please press OK. And see, the visualization button is active. Click on that. Yes. Okay. Right. So the immediate button that you need to click is end. Now, because you have to complete the whole stuff, right?
Yes, sir.
Yeah. Click on that. Excellent. Wonderful. Yeah. Click OK. So see, now your pipeline is complete. So your pipeline is complete. Now, since this is a demo pipeline, it did not take much time to complete. Now, if you like to visualize, click on the visualization button. So it will open a new window with JBrowse. So this will take some time for others. So can you, if you may stop sharing your screen and let others give.
So I had one query.
Yeah, please go ahead.
So actually, I tried to install the integrative genomics viewer, but it is not installing on my laptop.
Okay, so let me ask you, which version of Windows do you have?
So it's Windows 10.
Windows 10. And what is it a i10, i3 machine or a i5 machine?
So I'm not sure about that.
Okay, so is it a Pentium machine or is it i3 or i5?
So actually, I do not know. It's a Acer laptop.
It's okay. I don't. The laptop is fine. I also run my IGV in my laptop as well. But what you need to understand is, so is it a 32-bit machine or a 64-bit machine?
So 64.
64-bit. Excellent. So if you can go to the IGV page right now, I will be able to show it to you. Can you go to the IGV page?
Yes, I'll try to.
Sure. Now open a new window. Yeah. So I just take me to the place from where you tried to download IGV.
Yes, IGV. So from this software.broad institute.
Yes, that is the place. This is the place. This is the place.
So from here I did IGV for Windows.
Yes. You should click on that. Perfect. Yeah, click on that.
So I clicked, but nothing is happening.
No. Yes, click on that. Click on that. Yes, see, this is downloading. See?
Yes, it's 2 minutes.
So it's 41 MB. It's a pretty big file. Okay.
Yes.
So let the thing get downloaded first. It will take some time.
Yes.
See, it takes, although it says 2 minutes. Now, maybe I can get back to you once again during this course of lecture and show it to you how to install IGV.
Okay.
Okay, Devalina?
Yes, sir.
Great. Any questions you have?
No, sir. I had a query regarding factor regression analysis. Like I was
Okay.
trying to do it on the server, but I cannot
Mm-hmm.
like process it.
Okay. Can you show me how did you do it? How did you try to do it? Can you go to the server and show it to me?
No, sir. I have a query from the educational platform regarding this factor
Great.
regression.
Yeah, I'm there to answer your queries. All your queries will be answered.
So actually,
Let me.
there was one lecture in which it was shown that how to form the table for factor regression analysis.
Mm-hmm.
I watched the video that the link was posted, YouTube link,
Mm-hmm.
regarding how to make the table, but I could not understand where the from where the data like it was obtained.
Can you show me the video right now? Can you go to that page?
Pratin,
Yes.
if I may recommend, maybe yeah, Shubhrajit can show factor regression once.
Yeah, sure. Why not? Yeah, Shubhrajit,
Rather,
are you
that
there?
would.
Yes, sir.
Please help Devalina out here.
Yeah. So, could you repeat your query once more?
Uh, yes, like factor regression analysis, there was a lecture within the course, Transcriptomics 2.
Yeah.
So in that, there was a table that we had to create, and I could not understand from where the data I have to get for the table.
Okay. So, for the
So can you
factor,
go there, Devalina, so
Yes,
that
sir.
Shubhrajit
I'm
will be able to
trying to
help you
open that.
properly? Yeah, please. Yeah.
so basically for factor regression analysis, you have to upload a file. So that file which you have to upload is a RSAM expression table or an abundance table which you are getting from a RNA-seq pipeline. Now, when you are getting the result, you have to download a particular result or a particular file. That particular file you have to open using Excel or any other spreadsheet. That's the standard procedure. But now let's go and see what you are precisely wanting to know.
Shubhrajit, when you are going to show the RNA-seq pipeline, maybe
Yes,
you can get back to Devalina once again.
yes, sure. I
So
will.
this is the video I was talking about.
Yeah,
Okay. Can you play the video?
Yes, sir.
yeah.
Yes.
Here, this is a standard RNA-seq pipeline result. Okay.
Yes, sir.
So here you can see there are various pipelines. One says expression genes ftkm.txt. That one is the RSAM expression table.
Yes, sir. But where can I get these files? Like, which is the data that I have to upload?
Yeah, for the factor regression analysis, you have to upload a quantile normalized file, which I will be showing you during the workshop. So once I follow the entire procedure stepwise, then I think you will get a better understanding.
Okay, so I have run the previous pipelines as it was given in the lecture,
Yeah.
and so from there I had got one file which I had to normalize. Like
Yes.
when the quantile normalization was taught in the lecture, so
Yes.
there I had got one file which was a normalized file, the .txt file.
Yeah.
Do I have to upload that?
Yes, you have to upload that in the factor regression analysis pipeline.
But so with that, this similar data will not come.
Actually, see the similar data here, the data which has already been uploaded, that one has been prepared in such a way that you can get this particular colored visualization. But that doesn't mean that if you upload your file which you have got from a content normalization pipeline, will give you some other result. Okay, so
So
you
that
have to.
means that I have to like it's okay that I get a different result and not this table.
Yeah, because the file input might change, the scenario might change, but the algorithm remains the same. So when you're running factor regression analysis, you won't be doing it just for one particular file, right? You will
No,
be
sir,
doing
right.
it for various analysis of yours. So you must know that what this algorithm is doing and what type of result it is producing in general, so that you can apply that knowledge for any file which you have to deal ever, right?
Okay, sir. So this is just an example.
Yes, this is just an example,
Okay,
and
so then that means for this, the example which is shown this table, we do not have the data for this table right now.
yeah, the data for this particular table you will get from this course.
Okay.
Here it's already written, right? You already should have done the pipeline and you have that file expression, PDX expression, right?
Yes, sir.
Yeah, so once you're going through this course, they're actually following a logical sequence. So they are dealing with one particular type of file, they are playing around with the particular file. Okay.
Okay. So I had downloaded this table, this PDF expression genes, and
Yeah.
I saw it in Excel.
Yeah.
So it looked like this, but I thought that I have to maybe create this table, and so I couldn't find the data.
No. You can either work with this data which is already uploaded in the educational platform, or you can go by the files which we have been using in our classes, and we have got as a result of the quantile normalization pipeline, and use that as well.
Okay, sir.
It's all right. You have to just correlate to every file. You have to understand the algorithm and correlate. It's not necessary that every file or output will look the same.
Okay, sir.
Okay.
Okay. Thank you, sir.
All right.
Thank you, Shubhrajit. Thank you. So yes, Devalina, I think you got your answer to your question.
Yes, sir.
And Shubhrajit will be showing you today about generating this file from he'll be showing you how to generate these differential gene expression file from the RNA-seq pipeline, and also he'll be showing you quantile normalization and everything, so you can have a more greater better outlook of the problem.
Yes, sir.
Okay.
Yes, sir.
So
So the IGV is already installed.
no, it has been downloaded now, so you have to install it.
Yes,
So
sir.
click on that, click on the up arrow, click on that up arrow. Yes,
So it
show in,
says
no,
okay.
show in the folder, show in the folder. Yeah, so it will open a folder. It will take some time, I guess. Yeah, click on that folder icon. Yeah. So it has been downloaded in your download folder. Go to the yes, go to the download folder. Can you see the file which has been downloaded?
This one is slow.
Yes, so click on that. Double click on that. Yes. It seems that you have already downloaded that file somewhere.
Yes, sir, but it was not working, so I just
Let's see how it works. So Devalina, you belong to Amity?
No, sir. I am a student of Techno India University, Salt Lake, 2016 batch.
Ah, okay, so you're a BTech in biotechnology?
No, sir. I am MSc in biotechnology.
Excellent. So excellent, yeah.
So the screen went blank. I don't know.
Don't worry, it will take some time. So what is the size of your RAM? Do you know that? 4 GB, 8 GB?
No, sir.
You have no idea. Okay. Uh, okay, let us do this way. Yes. So let us do this way. I think it will take some time for you to install the IGB. So in the meantime, can I go back to other people who needs help?
Yes, sir. Sure.
Sure. Can you stop share right now? We will get back to you, Devalina, and follow the procedure that Shubhrajit is going to show you.
Yes, sir. Definitely.
Okay. So can you stop sharing and maybe give the stage to Diksha?
Yes, sir. I'll do that.
Thank you, Devalina.
Thank
And
you so much.
I would request you to mute yourself at the same time. Thank you. Thank you.
Yes, sir.
So anybody else who wants to share their screen? So I think the next one in the line was Diksha. Diksha, are you there?
Thank you, sir.
No problem. Thank you. It's all my pleasure.
Sir, if you go to the chat,
Yeah, I
Sunita,
can see. I am seeing it right now. Yes. So Sunita seems to be ready. Yeah, Sunita, you can share your screen with us.
Yeah. Hello.
Yes, Sunita, can you share your screen?
Yes. 1 moment.
Please, thank you.
Okay, I'm sharing, but I still haven't gone to my.
No problem. So can you log on to the server?
Yeah. I think I need the email ID.
So
I need
yeah,
the credentials, right?
you need the credentials exactly. So if you go back to your yes, credentials and video. Yeah, this is the link. So you have to open the.
I think I and Apar had made some changes to my this thing. Let's see if it still works.
Let's see right now. So the server URL is server.t-bio.info. Excellent. Yeah. Yes. So log on. You already
I'm
logged on.
already logged in. Yeah.
Excellent. See, yeah. So let me know what you want to learn. I mean, you want
Yeah,
to.
that the variant calling is one thing that I want to begin with.
Sorry, Sunita.
The first the Jyoti was doing variant calling, right? Mutation.
Yes, she was doing
Yeah.
the variant calling and she was doing visualization. Do you want to understand visualization?
Actually, I know how to do this. Only I know how to do it for RNA-seq using
No,
the
today,
HiStat
now we
and
are no.
Sprint.
See,
I don't know
now we
anything
are,
more than that.
yeah. So now
I
we
actually
are.
don't know what I want. I don't know what I want.
No. It's okay. Don't worry. But today the workshop is based on genomics and transcriptomics, and as a part of that, I am showing you today the genomics pipeline. So the
Yes,
genomics
works
pipeline
for me.
here in our server is, if you go down, this is mutational variant parallel analysis of variant data. So there is a demo pipeline out there. Can you click on? No, not this one. Can you go back? Can you go back? Yeah. So click on the arrow just beside this line. Yes, click on that. Yeah, click on that. Exactly. So see, the demo pipeline means that the file which you require to run this sort of pipeline is already uploaded. So you don't have to bother about the input file right now. But let us go around and find out how to build a pipeline. So the immediate
Right.
button that is highlighted is start button. Can you see it on the left? Yes. Yes.
Okay.
So these are the things that we covered in the genomics thing. Can you see the writings, the pictures and the writing on the left?
Yeah.
This is called, we call it educational pop-up, because the educational pop-up tells you to what are there in these pipelines. So click on the OK button. This will take some time. So if it is not being done, anything is being done. So if you might like to refresh this page once again. So Diksha, okay, but if you're on your screen, if you're on your phone, then would be this is for Diksha. Diksha, if you're on your phone, would you be able to run the server? Because we are going to show you the server today. So I'll come back to you. Just a moment. Let me finish it up.
I don't know why it's not running.
No, it's okay. It will run. It will run. Just click on that. Yeah. Click on OK.
You are done.
So what happens is probably since you have kept the server open for a long time, that's why it is non-responsive. So if you can refresh that entire screen by the refresh button on the left.
Yeah, this one, right?
Yes, this one. Excellent. Press on the start. Yeah, click on OK. Okay, something is wrong here, I guess. So what I would suggest is that close down this window and open a new tab. This window and yes, open another window and write the credentials, server.t-bio.info. Yes, go down. And yes, press demo variant pipeline. Click on start. Yeah, okay. I don't know what is happening.
This is the issue I've been having.
So this is the issue you have been having all the time.
Yes.
So would you log out from your server again and log in? Yes, log out from your server. Signed out successfully. Now log in again. Yeah, so your credential are already there. Yes. Signed in successfully. Now go to the mutational variant pipeline. Yeah. The same problem, I think. So maybe, do you know how to clear your cache?
Yes.
Can you clear it? Can you close down this window? And yes, no, better.
I just close off everything.
Yeah. Don't worry. It happens. Don't worry, it happens.
This is where I'm always caught up.
Don't worry, yeah. Open a new Chrome.
Yeah.
Maybe Mohit can help you. Mohit uses a Mac. Mohit, can you help her to clean the cache?
If you think we are, I think we are going to waste time if we do this. We will do one thing. You go along ahead with others. I'll
Fine.
do this thing, and I have a career fair today, which is going to begin in 3 minutes.
Okay. Yeah, no problem. No problem.
Yeah, I'll finish off that quickly and try to be back
Sure.
if we are not done by then.
Absolutely. We are here till 9, definitely, for sure.
Okay, then. See you soon.
Thank you. Thank you, Sarita. Can you stop share and then so that other people can join? Thank you very much. So Diksha, I don't think you'll be able to share your screen because I know you are in your mobile phone. So maybe the next time we're going to have it. Anybody else who wants to share their screen? What about Prabir? What about Shtomalo? Swati? Velumani? Mubin?
Hello.
Yeah.
Hello.
Can you identify yourself?
Yeah, Prabir here.
Prabir, great. Do you want to share your screen and show us what is the problem, or if you want to tell us something about any of your query?
I want to share. Just one.
Sure, please. Go ahead.
I'm sorry to interrupt. Hi, Pratim. I will have to leave the meeting now.
No problem, Sunita. We'll share you the recording.
Okay. Thank you so much.
Yes, thank you very much.
Bye.
Thank you.
Is it okay now?
Yeah, Prabir. So yeah, I mean, you have almost, I think you have covered the mile. So you have already opened the. You have already opened
that
the yes.
Yeah.
So now if you can see on the left panel, the data that are there, this group 1 sample, 5765 is the cancer sample and 5766 is the normal sample. Now, what if you want to look at what is there in the cancer sample? So you have to select the checkbox beside group 1 sample _1. Yes.
Okay.
So something will, so once you click that, it will, yes. See, this is loading. It will take some time to load, don't worry. So Prabir, if I may know, where are you from?
Actually, I am from Odisha.
Great.
Now I am working as a postdoc in University of Kota, Canada.
Okay. So you're an assistant professor out there?
No, I am a postdoc.
Oh, excellent. Good to have you here, Prabir.
Okay, thank you so much.
No
but
problem.
Right now I came to my hometown. October, I am here.
Okay. So are you finding the course interesting?
Yes. It's interesting, quite interesting.
Yeah. So great. So see, typically JBrowse takes some time to load because what happens is by JBrowse we try to understand. So see on the left, there are 4 files: group 1, sample 1, SRR 9255765_1, and there are 2 samples on the left, 2 samples of cancer types, and 2 samples of normal types. So the second one, the first one is the normal sample, and the group 2 is the so this is the result of the variant calling pipeline, the variant calling pipeline that we did in the server. So if you have run the server, and now we want to visualize, we want to see that what are the variants that are there in the normal sample versus the tumor sample. So that is why JBrowse is used as a web-based form. And at the same time, if you want to really understand that what are the variants in detail, then the application that typically is used is called Integrative Genome Viewer. So Integrative Genome Viewer is an offline application from Broad Institute, which you can download and install in your machine. And the file that are analyzed by IGV is called VCF file, variant call format. Now, the variant call format file has to be generated from the pipeline itself. So let this thing load. If you can go back to the server, I can show you how to generate the VCF file. Yeah.
This one?
See, this is the visualization file, that visualization pipeline that you have run, which have used one algorithm specifically, BowTie2, which is a mapping algorithm. Now, let's say if you want to come up with a VCF generation file. So can you just go back? Can you scroll up a little bit, Prabir? Yes. So create a demo mute. See on the right tab, on the top, you can find 2 things: My Pipelines and Create Demo Mutation Variant Pipeline. Click on that.
This one?
Yes, click on that. Yes, exactly. You are right on there. Yes. So the immediate button which is highlighted is Start. Click on Start. Yes. Click on OK. Click on the OK. This is the educational pop-up. Yes. So the immediate button that you need to click is PCR Clean. Yes.
PCR clean?
Yeah, PCR Clean. Yes. Yeah. So what does PCR Clean does? It is explained in this educational pop-up. So click on OK. Yes. So again, BowTie2T is the next immediate highlighted button. That's the algorithm of the mapping algorithm. Click on that. Yes. So it explains what does BowTie2T does. So the immediate button that is highlighted is Strelka. Can you see differential variant pipeline? Yes. So Strelka is a interesting algorithm which tries to figure out. So click on that. You will get an educational pop-up. Yes. So Strelka is a method of somatic single nucleotide variant identification. So remember, in our genomic class, we tried to understand what are somatic mutations and what are germline mutations. So this tells you that why is it so important to figure out see, it tells you how to what exactly somatic Strelka does. Strelka is a very famous, pretty famous pipeline, a pretty famous algorithm in mutational variant section. So can you click OK? Yes. Yeah, it will take some time, don't worry. Yeah, so immediate button that is highlighted is somatic sniper. See on the yes. So this will identify the somatic variants. So somatic sniper employs a Bayesian comparison of genotype likelihoods and the associated publication. This is the open source algorithm. So if you want to study more about the algorithms, then you can go back to this paper, which is written by Lee et al. In 2008. And this is also a standalone software package which is being used. So click on OK, because our motive is to generate the VCF file. So the immediate button that is highlighted is End, if you can see. Yeah, go to the button. Yes. Correct. So now your pipeline is complete. But then you have to press OK to complete the pipeline. Yes. It's not working?
Yeah.
Yeah, now see, this is the pipeline we built. Okay, so this is the pipeline we built. Yes, so we used 4 algorithms out here. PCR clean, which cleans the artifacts of PCR reactions. I mean, the polymerase chain reaction artifacts, the amplifications and all those adapters are cleaned, etc. are clean, and therefore thereafter it is mapped onto the using the bowtie 2, and therefore we go on to use Strelka and somatic sniper. Now remember, this is a demo pipeline that you have run. Okay,
Yes.
so if you have today's presentation, there is a slide which says variant calling in cancer. Do you have that slide with you?
Where I'll get that?
No, I think we have sent an email to you. Can you open your email? I think it is in your email. So
Okay.
yeah, maybe we have sent, yes. Can you, the second email that you can see? Yes, yeah. So today's presentation, if you click on the click here button, you can find today's presentation. It will ask you to download. Don't worry, it will take some time. Yes, so it directs you to the Dropbox where the presentation is. So this is the presentation. This is loading out there. Yeah, it's in the form of a PDF. So you can download the PDF.
Okay.
So if you go back to the pipeline, I will show you where the VCF file has generated. Yeah. Yes. So can you go up a little bit? Can you see something called download pipeline? Go up, please, Prabir. Download pipeline. Yes. Click on that. Yes. So there is a zipped file, Strelka,
This one.
somatic sniper. Yes. Can you click on that?
Mhm.
It will be saved automatically to your machine. Yes. And first you have to unzip the whole thing. And then within that file, within that zipped file, you will find the somatic snipe, something called VCF. So let us wait till it's downloaded. Yeah. So go back. Yeah. Go forward. No, go forward in the, yeah. Yes. Can you go down a little bit? Yes. Go down, go down. Yeah. So there's a slide called mutational variant. So we were here. So this is what we talked about. Yes, this is the pipeline that you have run right now. Can you go down? This is another pipeline you can also run. So this is the slide I'm talking about.
Okay.
See, so there are 2 SRA files that you can see. Export data size service T22 SRR 925.765.SRA, which is the cancer sample, and SRR 925.5766.SRA is the normal sample. Now, the pipeline that you ran right now has used these 2 files to identify differential mutational variant. Okay, so if you go to the server, you can do similar things, but before running the actual pipeline, it is better to go to the demo to understand what are the algorithms that are typically used. So if you go to the server again, can you go to the server? Yes. So has your file been downloaded? So let it download, but go back to the home page. Click on the TBio platform.
This one?
Yeah, this one. So it will open the main page. See, now if you go to the mutational variant place, click on the main line, not the arrow. Yes, click on that. Great. See, now we have some input file to be uploaded. Now, see, there are 4 input types. One is type of reference genome, R model genome, GTF. Now, this is a model genome that we are working with. So we need not change anything else. The format of the NGS data out here is FASTQ. Can you click on FASTQ? There's an arrow. Yes, click on that. Yeah, FASTQ. So the type of file that you're going to upload is FASTQ. And there is another output called single end or pair end. So single end, this is a paired end read. So it has the DNA fragments which are being read are from both ends, from 3' to 5', and also from 5' to 3'. And the organism out here is human. So you have to click on that and select the 38th version of, yes, exactly, 38th version. Yes, that's the latest version. Now you have to upload the file. Can you go to the file upload? You can see the, yes, exactly. See, now you have a lot of options out here. So what we have out here is SVL. Can you see that SVL button checked? Blue one, the blue one? Yes. So write the one of the links, copy one of the links from the PDF. Go to the PDF of the presentation.
PDF. Okay.
Yeah. Yes. Exactly. So copy that, the first line. Yeah, on the left. Yes, this one. Not the cancer. Copy up till SRA. Nee, copy up till SRA. Yes. Yes.
This one?
Copy. Yeah, this one. I think the A is not highlighted. The A hasn't been copied. No. The link, you have to copy the link. Let me help you out. So are you can you access the chat? I will help you with the file. Don't worry. I'll help you with the chat. I will paste the thing that needs to be pasted out here. Exactly this one you need to paste. So you paste this one. I have posted in the chat. Can you go to your Zoom?
Yes.
No. Can you go to the Zoom meeting from where you shared the file? You can find something called chat out there. No, the Zoom meeting from where you have shared. From where we have shared the screen. The Zoom application. The Zoom application from where did you share the screen from? Yeah. Go to the Zoom. Go to your Zoom application. I can see the Zoom application on the right of your panel. Yeah, this is the one. Yes. So now, see, you have one SRA file which is a cancer sample, but we want to understand differential variation. So you need to click on the button on the right, the black button that you can see on the right. Where, yes, click on that. Yes. No. So go to the add file. Go to the add file. Not this one. Not this one. Not this one. The upload files. Add files. No. The URL bulk upload. The URL bulk upload. Can you see a button called URL bulk upload? Yes. Well, you can do it by that way.
So may I?
Sorry, Shubhrajit?
Sir, may I guide him?
Sure, absolutely.
So you have to click on add more, which is under files from link heading. Yeah, add more. So you will get one here, and you click on add more once again, you will get the second bar as well for the normal link. Okay. Yeah. So now you have got two. So now in one box each, you have to paste each of the sample, one for the cancer one and one for the normal one, which has pasted in the chat.
Okay. So this one.
Yeah. Okay. Just a second. I'll provide you the link.
Yeah, I have already given you the link in the chat, Praveer. Can you go to the chat?
Yes.
Yeah.
The 5766.sra. Yes. So now you have to do group selection. Can you do group selection now? The 3. Can you go down the yellow button? Yes. Go to that. Now you have to select the group. What are you trying to do? Now there are 2 sections: group treatment, group control. Group control will be the normal sample, and group treatment would be the cancer sample. What is the cancer sample out here? 6,5 is the cancer sample. So drag the 765, yes, drag it from there and put it in group treatment. And 6,6, drag it from, yes, exactly. So now continue. There's a continue button out there, yes. Now you can start. So groups have been added. See, it's written out there. Groups have been added. You can create a new pipeline. Now, the immediate button that is highlighted is start. Can you click on the start? See, now you are going to build a new pipeline. So what does it do? What does this pipeline, mutational variant pipeline do? This is explained out here in this chat, in this educational pop-up. Click OK. Yeah. So see what not you can do out here. See, you can variant call directly. You can do PCR clean. You can do bowtie2, etc., etc., etc. So if you want to do the somatic sniper and Strelka, so first you have to do PCR clean, then bowtie2, and so on and so forth. Now, if you want to understand what is Parnickel basic and Parnickel basic mutations or Parnickel basic junctions, these are something different. Now, let us not go into those intricacies right now. Let us try to understand how a basic pipeline is built. If you click on PCR clean now you're building a new pipeline.
Okay.
Completely new pipeline, which is built by you yourself. Yeah. Click on save. Yeah. So you can run you can select two algorithms right now, either BWA or bowtie2. Now we usually select bowtie2 because bowtie2 is is a better algorithm in terms of mapping. Now click on that. Click on save. See, now you can do so many things. You can do VCF so variant calling. So now we are trying to understand variant calling or visualization. See, there are 3 columns where several buttons are highlighted. You can see the visualization button highlighted. You can see the Parnickel basic mutations, Parnickel basic junctions highlighted in the variant calling section. There is different diffchi algorithm highlighted in the differential variant calling button on the differential variant calling panel. There is diffchi norm. There is Strelka. There is something else, and I think the somatic sniper will be highlighted only after you select something out here. So the best thing to start with is obviously, Strelka. Strelka will give you the VCF file, how to generate the VCF file. Now, can you click on Strelka? Yes. Yeah. Click on save. So now you are building a pipeline by yourself. So the immediate button that is highlighted is somatic sniper. You can run several algorithms. See, you can run several algorithms in this differential variant calling section. So you can run Strelka, you can run diffchi, you can write diffchi norm. But the usual pipeline pathway that we take up is Strelka. We can end the pipeline out here by by clicking the end, or you can go on to select the somatic sniper also.
Okay. So the somatic
Yeah, click on somatic sniper. Yeah. So minimum depth is 10. That is the default value that we like to keep right now. Now, what does this somatic sniper does is explicitly explained in this educational pop-up out here. Maybe when you run it for the second time, you would like to read about it and then try to understand what all you are doing. So this is basically the tumor BAM file. That's what we have uploaded. Normal BAM file. That's what we have uploaded. We are calling, we are using the algorithm called variant called somatic sniper. Now we would, at the end of our operations, we will get something called variant.vcf. Now click on save. Yes. So now let us end the pipeline right now. Let's run it, make it simple. Let's keep it simple right now. Yeah. So save. Do save. Do press save. Now you have to name your pipeline. So give a name to your pipeline. Let's say, whatever you want to give. something which is small and understandable for you. Give it a name. Yeah.
Mine?
Yeah, give it a name. Say, let's say diff-sec probir. You can give it a name like that so that you can identify. Yeah, perfect. Mutation, yes. Right. Now click on save. Right. So now your pipeline has, see, your pipeline has been built now. Now you have to run the pipeline. See, there's a run pipeline button, big button on the left. Yes, click on that. Unless and until you run this pipeline button, press this pipeline button, the pipeline won't start. See, you have already started your pipeline. 1% pipeline has started. Now, if you want to recheck whether your pipeline has really started or not, you have to go to My Pipeline tab. There's a My Pipeline tab on the left, on the top. Can you see it? No.
Yes.
Yes. Click on that. Yeah. So this is the pipeline which is already run. So it has started, and at the end of this thing, you will get a VCF file. Now I think your VCF file is already downloaded, the one that you downloaded from the demo. Yeah, can you click on that? Can you open that zip file?
Yes.
Yeah.
One.
So, yeah,
Excel sheet is
exactly.
there.
Yes. Can you double click on the, yes. So you have two files out here. See, cell line somatic sniper header.xls, and you have a VCF file. You open that VCF file via the integrative genome viewer that is there if you have installed it. So the header, the somatic sniper header, if you click on that, that's a spreadsheet file. If you can open it, I can show it to you. So this contains the Excel information, the information about all the variants. Yes, see, there are so many information. So you can find what are so on the left panel, sorry, on the, if you look at the C column number C, so that's the residue in the reference genome, and what are the differences that has been noticed. See, originally in the so the one which are highlighted are kind of constant places, but there are a lot of mutations, as you can see out here. So G has been replaced by, this is the Excel file of the information which is there in the VCF. Now it is impossible to figure out what are the mutations that are there. There's another tab, if you can see on the bottom, are called key substitutions. Yeah. If you go to the left, can you see the substitutions out here? Yes. So these are the substitutions that has been noticed in the two normal and cancer files that has been uploaded into the server. So study about it, Prabir. I think this is a good start for you. And let the VCF file get completed, your pipeline get completed, and then let us move on, because we have to cover, we don't have much time left today. So let us go to some other participant, if we may. Thank you, Prabir.
Yes. Yes. Thank you.
Thank you. Get back to us if you have any queries. Okay.
Yes. Sure.
Thank you. Thank you, Prabir. Yeah. Can you stop sharing the screen? Yes.
Also, sir, may I just address one question which
Sure,
was
sure, absolutely.
Okay, well, asked by value. I hope you saw whatever Pradeep demonstrated using Praveer's screen. I hope you noticed one thing, that when the main pipeline was being built, even after clicking on PCR clean and POTI2, we got the visualization tab highlighted, right? Which was earlier not available in the demo pipeline, and that was your question. So the explanation to this is, the demo pipeline is like the training wheels, where we have already provided you a very few options. The entire field is not available for you, but in the main pipeline it is obviously available. So I think that answers your questions, that why the visualization was not available while you are clicking or going through PCR clean. The answer is, since it was a demo pipeline. Thank you, sir.
I think it's time for you to share your screen, and Prabir, you should stop sharing your screen so that Shubhrajit can share it, and You can go ahead and show the RNA-seq maybe. We don't have much time left.
Yeah, sure.
Perfect.
So,
and thanks
the
for
trans-
great. Thank you, sure.
Yeah, thank you so much. So the transcriptomics section was already covered in a few classes back, where we had talked about the RNA-seq pipeline, which is one of the most basic pipelines available on a server platform. Here are the four major areas or the sections of which an RNA-seq pipeline is built. The first one is pre-processing, where we use two algorithms known as Chemomatic and PCR clean. Chemomatic's function is to remove the adapters, which are known as the technical sequences as well, which are used for next generation sequencing. So Chemomatic trims them off. PCR clean is an algorithm which removes the highly expressed unwanted PCR replicates which were formed during bridge amplification or bridge PCR, which is a step of Illumina sequencing. So if you read through the procedure of Illumina sequencing, you'll be able to know more about that. The next function which an RNA-seq pipeline does is mapping. Mapping against two things. One is a reference genome and another one is mapping against transcriptome. So we have different algorithms, namely bowtie2G, top hat or HiSat2 for mapping against genome and bowtie2T for mapping against the transcriptome. The other thing or the algorithm which follows all these mapping algorithms is known as an RSEM table expression, or which is otherwise known as quantification or abundance table production. What it does is it produces a table which shows how much expressed is your particular RNA in a particular sample. We can build a pipeline till this, where you get your expression table and do further analysis like PCA and factor regression analysis, or we can go one step further and perform a differential expression analysis using DEC2 or HR algorithm. So what a differential expression pipeline tells us, we will be covering today, and I would like to show you what differential expression actually means. So this is the pipeline which we will be using. I hope you all have gone through the demo pipeline of RNA-seq, which is available on the server platform. But today I will be showing you how a real pipeline looks like, and we'll try to analyze how to do it, right? So here is a server platform, the bioinfo. Under the NGS data section, you can see a header called transcriptomics. So now we will be dealing with transcriptomics, and the first one is RNA-seq. So we will be clicking on that. We won't be clicking on the arrow and going to the demo one, but we will be building a real pipeline. So as you can see, again, we have an option called preliminary, where we have 4 areas which we can set. The first one is the type of reference genome. We will be keeping it as R model genome GTF. Reference model genome gene transfer format is the format which we will be selecting for this one. Format of NGS data will be FASTQ. We have 4 types of formats: FASTA, FASTQ, BAM, SAM. We will be using FASTQ in this particular pipeline. Next one is single air or paired end reads. We will be doing it as paired end. And finally is the organism of whose data we are trying to analyze. So we have to change it and set it to Homo sapiens GRCH38, which is the latest version of human genome, and then click on file upload. So here we have got the area where we have to upload the file. So now the question arises, from where can we get the file? So best place where we can get the file actually is Transcriptomics 1 course. So if you go to the Transcriptomics 1 course, and you can see on the left hand panel there, you can see the courses. If you go to the second one, 2.2, you will see a simple example of an RNA-seq analysis is the heading of this particular section. And if you scroll down, you will see here you get an option called download breast cancer SVL. So let me click on this and let me download this particular SVL file. Now what does SVL file mean? SVL file means that you have a very huge amount of data which you cannot upload from your end. So what PineBiotic does is it already uploads this file in a particular server which they have, and makes an SVL link to that, so that you can easily upload bulk from your own system. So I got a zip file as named as breast cancer education 1 SVL. When I click and open the zip file, I see that there is one file known as breast cancer education 1 SVL. We need to export it, so I'm exporting it, and here it is ready. The type you can see is called SVL file. So now let's get back to our platform, and let's get back to the server. Here there are 4 different types of uploading file option. First one is known as upload files, from where you can upload text file or files with the extension of .txt. The next one is files from link, which just now you have seen in the genomics pipeline, where we were adding SVL links of cancer and the normal. So you have seen that. Next one is NCBI run table upload. You can upload tables from NCBI itself. And the fourth one is URL bulk upload. And here it's written upload.txt or .svl file to bulk import URLs. So you can use this button or this option to upload text files which are very huge in size, or SVL files or SVL links to the files which are very bulky in nature. So click on this, and you can see here the SVL file which we had just downloaded, and you can see the size is just 2 KB. That's hilarious, but actually the file it contains inside is massive. Once it's uploaded, we have to click on group selection. So now you can see that there are various files present here. But if you see the names, there's ER, and in the bottom we can see TN, TN, TN. So these names represent the 2 different subtypes of breast cancer, namely estrogen receptor positive and triple negative. So now since there are 2 different types of files, we will be adding one more group here by this plus arrow, group B, and we will be segregating this by drag and drop method into 2 different groups. Let's put TN into group B and ER into group A. So let's do that. All the TNs into group B one by one. Let's go on doing it. I hope you would also replicate this process along with me, because that would be real fun to do this. So I have already dragged and dropped every TN file into my group B, and now it's time for the ER ones into group A. So I have done segregating. Let's have a look at the chat. Okay, thank you so much, Pratim sir. So now I have uploaded and segregated all the files here. So now let's click on contrasts. If you click on this button, you will see there are various parameters like 11100000011 minus 13. We have to click on select all because we want to see all the contrasts available in our data, which we are going to analyze for differential expression, and then click on submit all changes. So now you can see it's written contrast selected. You can create a new pipeline. So now it's time to build our own differential expression pipeline. The first algorithm which you can see highlighted in yellow is start. Let's click on start. Click on OK. The educational pop-up tells you about RNA sequencing. What is it? What it deals with. The next part, as I showed you in the presentation, is pre-processing. And under pre-processing, we will be using first Genomics, which removes the adapter. You remember? Here you can see the diagram shows you just that, how it is removing the adapter from the sequences. Adapters are necessary for sequencing, but after that, they are not required for the data analysis. So once we have removed, clicked on Genomics, let's click on PCR clean, which removes the highly expressed and unwanted PCR replicates. Once we have clicked on PCR clean, we will be going to mapping on genome part, because the next part was mapping. So we will click on HiSat2. That's an algorithm which maps your reads to a reference genome. You can see here, it's mapping against a reference genome. There's a publication associated with it as well, so you can read it, get a better grip on it. Next, we would like to click on the highlighted button, which is already highlighted as cufflinks. You can see on top of cufflinks, it shows isoform construction. So what are isoforms actually? You all are familiar with splicing. That's a very major event which occurs in eukaryotes. So when the splicing event is occurring or the introns are being removed, they do not happen at the same position every time. It can vary. So once it starts varying, various different types of spliced products are produced. Those are called isoforms, and this process is known as alternative splicing. So when you are mapping against the transcripts, you are not only mapping against one transcript, but you are looking at every possible transcript or every possible isoform. So that's what is known as isoform construction, and that's what cufflinks does. If you read through this, you will be getting a better understanding how it maps your reads against all the transcripts or all the isoforms present. Once this mapping and isoform construction is done, the next one is cuff merge, because it will merge all this transcript or isoform mapped and convert it into a GTF or gene transfer format file, which can be further used by Botai2T for transcript mapping and for production of expression or abundance table by RSEM, which is our next algorithm. So first we will map on transcript using Botai2. There's BWAT available as well, but Botai2T is more popular and trusted. So here you can see how it does an ungapped multi-seed alignment for a very huge amount of reads. So let's click on save, and now let's go to RSEM expression table. What RSEM expression table does is it follows the alignment done by Botai2T and finds out the abundance expression or expression level or abundance for a particular transcript or a particular read. On the right hand side, you can see a parameters option. There are 3 parameters available: FPKM, expected count, and TPM. These are all types of expression formatting. So we will be using FPKM, that's fragments per kilobase million. You can go through this FPKM, RPKM, or TPM, and you can understand what it actually does. After we have generated or clicked on the RSEM expression table, we will be clicking or going to the next part, which is differential expression. There are 2 algorithms. One is HR and the other one is DESeq, and its new variant called DESeq2. We will be using DESeq2 because that produces a more accurate result. And once we have clicked on DESeq2, now we will be clicking on end, and that's the last algorithm, and that's how you have prepared a differential expression pipeline. Now it's time to name your pipeline. Let us name it. That's how I name it. And now let's click on run pipeline, and you can see that my pipeline will be off and running shortly. So I hope every one of you have more or less understood how to build this pipeline, and I think you can reproduce what I did. So yeah, my pipeline is running. You can see it's showing 1% started, and you will be also able to see how much space it uses. But the problem is this pipeline will take around 1 day. So it will take around 1 day to run. So I cannot wait for the output files for that long. What I will do is I'll show you how the output files looks like. Okay, so this is how the result will look like once the pipeline is done with running. There will be a plethora of results or output files available, from which we require just 2 files. I have shown it in the arrows. The first one is DESeq2 allfiles.zip, which will give me the differential expression result, and the other one is expression genes fpkm.txt. That will be giving me the abundance table. So now let's go to the files. I have already downloaded these files and kept it ready. So for analyzing these and visualizing these, we will be having to use MS Excel or any other spreadsheet program. So let's open Excel. Once you have opened Excel, you have to go to this data tab. You can see here, and then click on from text, because your data is in text format. Which one? The one which says expression genes fpkm.txt. You can see here this file says expression genes fpkm. So we will double click on that. Whatever parameters come up, just select next, finish, and okay, it will be a tab delimited format. And here is what we say as an abundance table. You can see on the first column, these are the gene IDs, gene names, and on the column B, C, D, these are the sample IDs. And for a particular gene, this is the particular amount of its expression in this particular sample. This one is for the second one, this one is for the third. But I think you can see a problem here. The problem, if you notice, is for the first gene, it shows 8.11, 1.54, 9.27. But for the second one, it shows 41.19. And if you go to this one, you can see that it shows 166.37. If you go further, you will see there are much bigger values like 193. So don't you think, and here I can see it's 0.64. So don't you think it is similar, like measuring something in terms of kilometers and miles? They are not in the same scale. So what we need to do is we need to first of all bring this expression into a similar scale format. So for that, what we do is we have to go to the server platform once again. But this time we will be going to the data integration and modeling area. And from there, under data mining section, we will go to utilities. In utilities, we will get the option to file upload. Here we will be uploading that particular file which we got as a result. Which one? This one, which says expression genes FPKM. Let's upload it. What I'm going to do here is I will show you in the presentation. I'm going to build this. This is a utilities platform present in the server platform where I will be doing a log transformation. Now, what is log transformation? The log transformation algorithm will transform every expression value logarithmically and thus bringing each and every expression into the same scale. And once it is done in the same scale, we will be getting a very better understanding, and also we can compare easily the expression levels of the different samples or the different genes in different samples. So let's get back the platform. Sorry, my screen got unshared. Okay. Yeah, let's go back to the platform. My file is uploaded. Let's click on continue. We will click on start. Here's the utility section. We'll click on quantile normalization. And here you can see the option comes transform table to LN, logarithmic number. Yes. For this particular pipeline, I won't be setting any threshold, so let's keep it 0, 0. I think you have already gone through this threshold part when PCA was being taught, but for this particular one, we will be keeping it as 0. Click on save, and directly let's click on end. Now I have to name it. And once we are done with the naming, we'll run the pipeline. This will be done pretty quickly, so I'll be waiting for this output. Meanwhile, let's go back here and let's have a look at the other file which we downloaded. That was deseq2allfiles.zip. So let's click on that, and here we have again a number of files. From here, what we need is this one, expression genes deseq2.txt. This is the file which will show me the differential expression from the same expression genes FPKM file which we used for our abundance table. So we extract that. Here it is, it's extracted. Now let's go back to Excel. Let's open a new tab or a new sheet and again import that data. So here we have expression genes FPKM deseq2. Let's import it. Let's click on next, finish, and again a similar tab delimited format table will come up. Now you can see the same gene IDs on the first column. If I may show you, see, it's the same gene IDs. But on the other columns, we have base mean, log to fold change, p-value, p-adjusted value. What is p-value? I think it has already been covered, null hypothesis and p-value. You know, we generally set a standard threshold of 0.05 or less than 0.05 p-value filter. Similarly, we do it for p-adjusted value. What is p-adjusted value? It represents false discovery rate, which is also known as Benjaminian Hodgkin principle. What it does is it is an iterative or a multiple hit p-value. So let's put a filter of less than 0.05 for p-adjusted value. Let's go to number filters and put less than 0.05 and click on OK. As soon as we do that, you can see many of the genes have vanished. As you can see in the IDs, it's after 1 it's 14, then 43, then 63. So you can understand many of them have vanished. The next filter which we would need to apply is the log to fold change, because we would want to focus on only those genes which have a particular threshold fold change or higher. For our particular analysis, let's put it between 1 and -1, which is a very stringent filter actually. But okay, let's do that. Let's click on between. First one is greater than or equal to 1. Then a very important thing, check on or. Don't do it and. If you do it on and, then everything will go blank. So click on or, and then is less than or equal to -1. Once we have done that, click on OK. So now many other genes vanishes as well. So we have a very small amount left right now. Okay. Now what this p-adjusted value does is, p-adjusted value also says that if you remember, we had two different groups of samples. One was ER and one was TN. So the smaller the value of p-adjusted value, the more expressed will be the group A first as compared to group B. So if you remember, in group A we had put ER, and in group B we had put TN. So if we sort the p-adjusted value in such a way that it's in ascending order, smallest to largest, then the ER will have a higher expression and the TN will have a lower. And if we do the opposite, then it will be vice versa. So let's sort it from A to Z, which says that it's from smaller to bigger. And let's select the first gene which has appeared. Let's just copy this name, go to our previous abundance table and do a find option. Yeah, we have found it. So now let's select all the expressions and click on insert. And what I will be doing is I will be making a bar chart. Yeah, so you can see that this is my bar chart for the particular gene ID you can see here. So the first 5 samples were ER, sorry. So they have a very high expression as compared to 6, 7, 8, 9, 10, 11, which were TN, right? And that's what I told you just now. The differential expression actually does this. But there's a problem once again. The problem is, you can see this one is slightly over 3000 and this one is 7000, whereas this one is just below 2000. So again, this is not so good for an analysis because we don't know how much of a difference should be considered to be a very good difference. So what can help us out from here? The thing which can help us out is this one, which we had set to cook. That's called log transform. Yeah, my pipeline is complete. Let's click on download pipeline files, and here is the one which says the threshold 00 normalized txt. So this is my log transform normalized file, and we will be downloading it, saving it. Okay, it's done. Let's go to Excel, open a new sheet, and we will import this particular log transform and normalized file. This is the one. So here you can see every file now has an expression level of 2, 3, 1, 4, 0 point something. So see, all those hundreds and 40s have now been transformed to 2, 3s in a logarithmic fashion without the loss of any data. And that's a very important line. There's no data lost. So what we will do now is we will again click and copy this particular gene name. And now let's see what expression level does it shows in this particular file, which is already log transformed. We will perform the same thing. We'll copy everyone, go to insert, select on bar chart, and now you can see that on the left hand side, on the y-axis, it shows 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and no 7000, 13,000 is there anymore. And when we have got this kind of a plot, a difference of 2 or more is considered to be a significant difference, because log fold change 2 is a very high difference. So now we can say that, okay, so this is not such a great difference, because it's not even 1 difference, right? Or slightly closer to 1, which we weren't able to say when we had got a plot like this. See, here it's 3000 and it's here 7000, so we would tend to think that it's a very good difference. But now we can see that, okay, after doing the log transformation, it's not. So the another thing let's show is, what if we reverse the orientation of the p-adjusted value? Let's do it from sort Z to A. So in descending order. Now what we get as a first gene, let's copy it, go to our log transformed and normalized sheet, and again do a find of option. There you go. Let's copy it and let's do a bar plot. And that's what I said. When we organize or orient our p-adjusted value in a descending order, we will get those genes which are more highly expressed in the second group or the TN group as compared to the first group or the ER group, right? So here, if you look at the 6 and 7, this does show a higher than a log 2 difference. So we can see, yeah, that's a significant difference. And if you compare in the first group versus second group, then obviously it's a higher difference, very high difference. And that's why it came up as the first differentially expressed gene top of the list in our DE-seq2 result, right? So I think you have got an idea of how to analyze differential expression. For understanding more, I would recommend you to go to Transcriptomics 1 course and also Transcriptomics 2. These courses will give you a very lucid and very detailed explanation of everything which I had talked about here. And I think till DE-seq2 is a good thing for today. Factor regression analysis and other things we will be covering in the next workshop, I hope. So would anyone like to say anything, ask any questions, or if you want to share your screen and want to do a hands-on on what I did? So would anyone like to say anything? You can go ahead if you have any questions.
So anybody have any questions for Subhrajit? Wonderful, Subhrajit. That was sufficiently clear, I think, for the people. So anybody have any questions? I think you have explained it so well. People are, yeah, see, Velu is so satisfied. Yeah,
okay.
all praise to Velu, to Subhrajit as well, for it appears fine, I could create a pipeline. Good, good. That's what we want, That's what we want from you people, to create your own pipeline and figure it out, so that it will be a lot of help when it comes to getting your projects done at the end of the day, at the end of this course.
Yeah, because we always believe that we all must do. We just not simply sit and watch something. It's something which we do by our own hands. We cook our food and then we eat. Okay, so I think if there's no more queries, so I will transfer the stage to Pratim, and I will stop sharing. Pratim, sir, it's all yours now.
Thank you, Subhrajit. So I have pasted the thing that remains to be done on my part is not much. I think you should be going to the genomics platform and try to figure out what are there. I think we should meet more often in this sort of workshop, and so that we can have a more clear understanding about how to do stuff like that. So let me share my screen again and then show you what I have to share. I think people can see my screen. So I have pasted a link in the chat about a feedback form. This is something which we would like you to fill up, all of you, so that we get an idea about the kind of feeling that you are having about the material that we are covering, the type of delivery that we have, the type of server that you are using at length, etc. So it is very important for you guys to fill up the feedback. So we have covered many things about genomics and transcriptomics a lot today. And I think it was a very good transcriptomics session. And I urge you to complete the transcriptomics 1 and 2, and also the genomics, introduction to genomics and genomics 1 at the same time. So the next session, which is going to take place, feedback is very important, and please follow the link that I have shared with you guys, so that you can fill up the feedback, and then we will be able to know that what are your problems. Okay, and we would like to organize more of these sort of webinars via Zoom place. And maybe, you know, you should be, I think, you know, the way we are trying to do it is basically trying to help you out in a hands-on way to make your own analysis and research useful. And we would like to share screens like that, the way we did it today, and also the try to understand what kind of pipelines you're running, and also help you out to run the pipeline at your own.
Pratin sir, Debanita is having some issues with somatic sniper.
Okay, so let us go back to the chat. And unable to open the Strelka somatic. Okay, Debanita, the file is a zipped file, you know. So first you have to unzip the file. So what you have to do, let me show you. If you can see my screen, I'll show you how to unzip it. So if you, okay, let me, I don't know, where is it? I don't know. So it will be a, yeah, see, if you can see my screen, let me zoom on. If you can see my screen, this is the somatic Strelka_somatic sniper.zip. So I am using a Linux system. So if you are using a Windows system, then if you right click on this file, see, I have right clicked on this file. So I have so many choices, right? Open archive manager, open with blah, blah, blah. So what I would do, I would extract. So if you extract, it will be unzipped at your own place. And if I extract, see, a file has been extracted. So what is that? It has a file that has been extracted. The file is somewhere up there. Yeah, this is the file that is extracted from the zip. So this is an unzipped file. So are you able to do it? I guess you'll be able to do it because it's a zip file. So you have to unzip it. So it's kind of compressed file that is being downloaded. And if you double click on this Strelka.somatic sniper, you will find two files. One is the cell line somatic sniper header.xls, which I showed Prabir. This contains all the information about the mutations that you have. And see, there are so many mutations. All the chromosomes are being covered. How many chromosomes are there, and it also have the positions. But it is difficult to find out from this particular file that where are the mutations have occurred. So this is not something like the RNA-seq pipeline results. So that is why we provide here. Now, if you have downloaded IGB, have been able to install IGB at your end, you can open the VCF file IGB viewer. So if the IGB place where you have downloaded, there are some files out there. If how to load the VCF file, it will be a lot of help to you. If you cannot, we can cover it more in our next session probably, or sometime when we have these webinars, online webinars. Does that answer your question, Devanita? I have Windows. Great. I think, yeah. Okay, sir. So you try it out. Let us know what has happened. It's not like that kind of, answering the question and getting everything. Are trying to keep all the questions that you are asking. We are trying to solve the problem your own end. This is something which is, so that is why I always try to reach out to you via WhatsApp or something like that. Please get back to us if you have any questions. So happy to help. So if there are no more questions, let me go back to the presentation. And feedback, please fill up the feedback form. I have shared that in the chat. So copy it and paste it in your browser right now. And before as soon as we end our class, I would request all of you to fill up the, whoever has not filled up, I have seen people have filled up, but whoever has not filled up, I would request you to fill it up, because it would help us to understand how you have understood. Because see, then we can only be able to cater you the right thing that you are looking for. The next class is on 17th, which is on probably on Tuesday, next Tuesday, and we would be entering metagenomics. That is a very interesting world to understand, and we would be running a few projects there. So before we embark upon this journey of metagenomics, I would request you to do the introduction to bioinformatics, do the reviews about your understanding about introduction to bioinformatics 1, and also do the transcriptomics 1 and transcriptomics 2. And thereafter, it will be easier for you to really cope with the entire progress of the. So thank you very much for attending. I think we have kind of sufficiently enlightened you in the process of thank you, Ruchika. So yeah, many people have already filled it up, but I'm talking about those who have not filled it up as yet. So please fill it up so that we can have this idea about what's going on at your end. So thank you. I guess no more questions are there. We have already overshoot the time. So let us catch up. Are you able to create? Okay, fine. Yeah, nice. So see, I mean, I think yeah. Thank you very much. So wonderful work, Shubhit. All of you deserve accolades. So let us meet on 27th again. Thank you very much.